So I'm going to talk about the structure sparse canonical correlation analysis. So it's a joint work with Jaime and Jaime Carbonell. So thanks a lot for the Vincentia tutorial, like the talk on the sparse PCA. So it's somehow related, but I mainly focus on the computational aspect instead of statistical analysis. So first, let us look at the motivating example on the genome-wide association study, the so-called GWAS, where the genome-wide association study is to study the association between the genetic variation and a phenotypic trait. So one of the most common genetic variation is so-called single nucleotide polymorphism, so-called SNP. So each SNP is a difference in the single nucleotide of an individual DNA sequence. For example, in the common people, if you have a CG here, but if it changes to TA, so it's called a SNP. And there's about 1 to 10 million SNPs in the entire human genome, but only a few thousand are related to a disease. So in the typical genome-wide association study, we want to study the association between the SNP and the phenotypic trait, for example, the symptoms of the disease. However, as you know, the relationship between the SNP and the phenotypic trait could be very complicated, and it's often very difficult to directly study their relationship. So a more common way to study that is that we introduce some kind of intermediate phenotypic trait, for example, the gene expression data. And we first study the relationship between the SNP and the gene expression data. So this step is so-called eQTL mapping. And from that, we can study the gene expression data, and for example, we can use the gene expression data to predict whether the people has the cancer or not. So this has been well studied, and today I will mainly focus on this part, so the eQTL mapping. So let us write down its formulae. So assume we have the Y is the gene expression data, and X is SNP data. So they are measured on the same set of samples, like N sample. And there are a number of there are P genes and the D SNPs. And in the biological study, both the number of genes and number of SNPs are much larger than the sample size N. So the traditional methods to study this include like hypothesis testing or marginal regression. And recently, the people start to use the multivariate sparse regression to study this problem, where they can formulate this problem by regressing the Y on the X, and put some sparsity regularization on your coefficient B. However, I think the main problem is that this formation is kind of unsymmetric. So here we want to study the association between the SNP and the gene expression data. So the association is kind of a symmetric way to study their relationship. But here it's unclear that why you regress Y on the X instead of X on the Y. And more importantly, for example, if you use L1 infinity or L1 to norm under this B matrix, so basically you select a few SNPs that can simultaneously, you hope that they can simultaneously explain all the gene expression data. So it somehow could be very difficult in the real biological study. So then we resort to another recently proposed work by Wheaton and Tipshani and Hasty in so-called sparse canonical correlation analysis, sparse CC. So it's a kind of more symmetric way to study this problem. So basically you find two sparse vectors, U and V. You project X on the U, Y on the V, and maximize their correlation. We do a bit of relaxation and can formulate this problem into this one. So you maximize the product between X, U, and Y, V. And since you want U and V to be sparse, you put the L1 norm on the U and V. And here we also constrain in the original CCA, they put that the L2 norm for U equals to 1 and the L2 norm for V equals to 1. However, as you know that if you constrain on the board, so it's kind of non-convex and makes this optimization very hard. So we relax it a bit to make this to just less than 1. And so this procedure, so here we only find the first the canonical vector pairs, U and V, but this procedure can be easily generalized to find the multiple canonical vectors. So they have been applied this procedure to study several biological studies and achieve very successful experimental results. But however, the main problem is that if you only use the simple L1 norm, it's very difficult to capture the rich structure information among genes or SNPs. As we know, the biological, for example, the gene SNP carry out some structure information and carry out very strong biological semantics. So it's very important to incorporate this prior structure knowledge. So how we incorporate that? So a common strategy is that we can use the structure penalty that has been proposed and studied in the last 5 years. So in general, we denote structure penalty as the omega U or omega V. And in today's talk, I will mainly focus on 2 structures. The first one is the pathway structure among genes, and second one is network structure among genes. So the pathway is a whole group of genes that correlated together, and they co-function in some biological process to perform certain functionality. So this one we can view as a group structure among genes. And second one is the network structure. For example, it can be obtained from the correlation network or gene regulatory network. So today I will mainly focus on the structure among the genes, but you can extend this to study structure among SNPs and or the structure on both genes and the SNPs. The structure sparse CCA can be formulated in this way, so it's very similar to the previous formation, but we only changed L1 norm on the V to be the structure penalty. So you can see that this one you have the maximization over both U and V. So a natural strategy to optimization it is to do an alternating procedure, where you first fix a V and maximize over U, and you fix U and optimize over V. And you iterate these 2 steps until convergence. However, since we might need to iterate this many times, and so we need the solver for each sub-problem to be highly very efficient and scalable. And for this one, since you only have the L1 norm, so there's a closed form solution for this optimization problem, and it's given in the Witten's paper. So if you're interested, you can go to that. But today we are mainly focused on this optimization problem. Since it's very difficult, we have a very structured penalty on the V, and this structure could be very complicated. But before I go into the details of the how to do the optimization, let's review some basic structures. The first one is the group Russell structure, so we can use it to do the pathway selection. So we assume there are P genes, and we partition into different groups. However, here the main difficulty is that the groups are highly overlapped together, since each gene can perform different functions in different pathways. The gene can appear in different pathways. So basically that means that for any 2 groups, GI and GJ, their overlap could be a non-empty set. But we still, under this setting, we want to achieve the group sparsity. That's the VG for each group goes to 0 altogether. So this can be achieved by this L2 norm on each group, and we put the weight for each group as the WG. And instead of using this simple L2 norm, we add a quadratic term here, the C over 2, the L2 norm of V squared. So basically, it can address the collinearity problem. So it's known as elastic net penalty. And as you know, the collinearity problem is happens a lot in the biological study, so that we add this quadratic penalty. And we can reformulate a bit of the optimization problem with respect to V. First, we change the max to the min, and so we put a minus sign here. So instead of solving this constraint form, we solve the regularization form here. We put this constraint to the regularization. And you know there's a 1-to-1 correspondence between C and C2 to the tau and C theta. And if you further do a bit reformulation, you can recognize it as the minimization of the loss function. It's 1 over 2 V minus beta squared, where beta is basically Y transpose X U. And this is the L2 norm, the group Russell penalty on each group. And here we also have a constraint. We want the L2 norm of the V is less than or equal to 1. So this is basically we want to solve this optimization efficiently. And however, the main challenge comes from the complicated structure penalty. The groups are overlapped together, and also instead of from the Russell problem, we have the both constraint and the both regularization and also a constraint. So the traditional method, like interior point method, can be easily applied, but it has very poor scalability. The core descent is a very powerful method. However, when you apply here, the groups are overlapping, so the optimization with respect to each group could be very difficult. So alternating direction and augmenting Lagrangian might be another possibility. However, basically there's no convergence rate guarantees. So the first order method becomes very popular nowadays. And for example, you can use the projected subgradient descent or the forward-backward splitting, so-called FOBAS, to solve this optimization problem. But basically, they have the convergence rate of 1 over t, where t number of iterations. And if you may think about some other like proximal gradient method or Nesterov method. So they belong to a whole class of accelerated gradient methods. However, since you have very complicated structure penalty, there's no closed form solution for the proximal operator. So it's not clear how to directly apply this method. So today our solution is that we first talk about we smooth this structure penalty and using the Nesterov smoothing technique. And then we specialize the so-called excessive gap method. So it's a primal dual method developed by Nesterov in 2005. And this can be achieved a convergence rate of 1/t squared, which is much faster than the 1/t. So let us first talk about how to smoothing the structure penalty. The key observation is that we do the smoothing using the dual norm. So here is our structure penalty. We know that the dual norm of the L2 norm is still L2 norm. So for each VG2 norm, we can write it as the maximization over L2 norm of alpha G and the inner product between alpha G and VG. So we replace this maximization term for every place, and so we replace this, the VG2 norm by this maximization form, and we do a bit reformulation. So you can write down as the maximization of alpha belongs to Q2 and CV and the inner product with alpha. So C here is a highly sparse matrix that indicates which variable belongs to which group. And so here we don't do any tricks. So here is just a reformation. So it's still a non-smooth function in V. And we apply Nesterov smoothing technique. So we minus a mu d alpha here to turn this non-smooth function into a smooth one. So mu here is a smoothing parameter, and d alpha in principle can be any positive value strongly convex function. But here we just choose 1/2 alpha 2 norm square for the simplicity. And if you are familiar with convex analysis, you immediately realize that it's just a functional conjugate of a strongly convex function. From a basic fact from a convex analysis, the functional conjugate of strongly convex function is smooth, so it's a smooth function. So we just turn this one into a smooth function. And so the smoothness is kind of controlled by this parameter mu. So I will give a geometric illustration to show this smooth approximation. So consider a very simple case where the PV is just the absolute value of the V, and V is just a real number. So this can be written as the maximization of alpha times V, where alpha belongs to -1 and 1. So it just corresponds to this blue line. You can see it's a kink here since we take the absolute value, so it's a non-smooth function. And we take the mu x to 1, we get this red line. Mu x to 2, we get this green line, and mu x to 4, we get this purple line. So as you can see, we just smooth this kink by this quadratic approximation. And for larger mu, we get a bad approximation, but for smaller, we get a good approximation. So the relationship between the smooth penalty and the original penalty can be easily characterized by this inequality, where the smooth penalty is the lower bound of our original one, since you minus mu d alpha here, and the gap is controlled by this mu d, where d is the maximization over this d alpha. So here it's very simple. It's just number of groups over 2 if you derive it a bit. So you may come a natural question that in practice, how do we choose this parameter mu? So as you can see, when the mu is large, you get very bad approximation. But it's leads to a small Lipschitz constant, so that the function shape is rather nice, so it's lead to faster convergence. But for a smaller mu, you get a good approximation, but convergence is kind of bad. So a natural strategy here, you're starting from a larger mu, so you get bad approximation, but since it's at the beginning, so it doesn't matter that a lot, and you gradually decrease your smoothing parameter mu. So I will verify this point in the later theoretical analysis of the convergence of excessive gap method. So then we have a smoother problem. So we change it using Nesterov smoothing technique. We just change P mu to be the structure penalty to be P mu V, and this inequality also holds here since we just do the substitution. Okay, now we just go on to the excessive gap method. So it's a primal dual method. We write down the primal form, and we can derive its dual form. And from the functional duality theorem, you know that the objective on the dual is always a lower bound for the objective on the primal. And some regularity conditions at the optimal, the mean of the primal is equal to the maximization of the dual. So the core idea of the excessive gap method is that it simultaneously updates the primal solution and the dual solution. And for each iteration, it keeps the key inequality that on the smooth objective on the VT is the lower bound of the dual objective. So from the functional duality theorem, we observe that the phi alpha t is the lower bound of FVT. And from the previous slides, we know that the FVT is upper bounded by the smooth approximation plus the mu TD, if you remember the inequality in the previous slides. So as you can see, if we're starting from a larger mu and gradually decrease it, for example, at the convergence, if mu t goes to 0, then mu t goes to 0, so everything will be equality. And then we have the phi alpha t equals to FVT, then we get to the convergence. So this is a high-level idea of the excessive gap method. And I will give a graphic illustration here. So this is your original objective FV, and this is its dual function. And at the point that the FV meets phi alpha, you get your optimal solution. We construct a smooth approximation, and the maximal gap is characterized by this mu TD. And for each time, we find 2 points. But we guarantee that F mu TVT is the lower bound of this phi alpha t. And of course, both of them are lower bound of FVT. So we gradually decrease this gap. So then we'll finally, this one will get closer and closer to your original objective, and you will hit this point. So the convergence rate here is very simple. So you can get the convergence rate of the primordial gap. It's FVT minus phi alpha t. And so basically you substitute this one here, and it's minus phi alpha t. And this will be upper bounded by this mu TD. And according to Nesterov paper, it's just the mu t is set to be t over t plus 2, the mu t minus 1. And expand this, you'll get this 1 over t squared. So the convergence rate is approximately d over t squared, which is much faster than the original projected subgradient descent, which 1 over t. And here the per-iteration complexity in our problem is very simple. So it's just the sum of number of groups. When you're applying the Nesterov method, you have to careful, since you have to compute the gradient of this is smooth approximation and the phi alpha here. But nice thing here that since we're using the d alpha to be 1 over 2 alpha 2 norm square, so all this gradient has a closed form solution. So they are deriving the paper, so you can go into the paper. So here I just highlight the idea of the excessive gap method. So we talk about how to do the optimization for the group case, and it can easily extend to the network structure penalty. So in the network structure penalty, we use the group fused penalty, use the fusion penalty. So here we assume we have a prior knowledge of the group structure on the genes, and if they are connected by the edge, we enforce their coefficients to be kind of similar. So this fusion penalty has been originally proposed by Tipshani. And we also put the L1 norm here since we want the V to be sparse. So this fusion penalty can be naturally used for the group pursuit purpose. We are here with group pursuit, we mean that we can learn the group structure using this fusion penalty. So remember previously we talked about how to incorporate the group structure, but the group structure is given as a prime information. But here we assume that we don't know the group structure, but we can using this fusion penalty to learn the group structure. So if you remember that in our original Tipshani paper, it's just to put the fusion penalty on the Vj minus 1 minus Vj, so it's a piecewise constant. So a piecewise constant can be naturally viewed as a grouping in a series order. And you can use a complete graph, then you do the pairwise grouping. And here we use the graph-guided grouping where you have a prior knowledge of the graph. If 2 vertices do not connect by an edge, so that we don't put a penalization on them, so they are likely do not in the same group. So optimization for this one is also very similar to the previous. So basically we do a bit reformulation. We can reformulate this one into CV, the L1 norm on CV, where C is the edge vertex incident matrix that indicates which vertex belongs to which edge. So as you know, the dual norm of L1 norm is L infinity norm. So it can be written as maximization of L infinity norm on alpha less than or equal to 1 CV times alpha. So it has exactly almost the same form as previous case. So we can introduce a smooth approximation and using the Nesterov method, excessive gap method to optimize it. So here we do some comparison of the speed. So here we only focus on the sub problem where we are optimizing with respect to V. And we compare with excessive gap method, interior point method, accelerated projected gradient descent, and also the alternating direction augmented Lagrangian. So as you can see, we vary the number of variables to be 40,000, 500,000, and 5 million. And for each time, we compare this 4 method, and we also vary the theta, so we choose different number of variables. So as you can see, in all these different metrics, our method is faster than these competitors. And even if the p goes to 5 milli, our method is kind of converging about 100 seconds, so about 1 or 2 minutes. So it's really fast. So yeah, so we also conduct some simulation study to verify the advantage of group structured sparse CCA. So basically, we adopt the experiment setting from the Witten's paper, and here is the ground truth for the U vector, and this is ground truth for the V vector, and the V is constructor of some overlapping groups. We randomly set a few groups to be 0, and for the other elements, we randomly draw from a Gaussian distribution. So if you apply the L1 sparse CCA, you can see you get the U almost okay, but in the V, it's kind of over-sparsified. You cannot capture this, the V. But if you incorporate the prior knowledge of the group structure, you will recover this V much better. So you can see here. And also, we use the network structure to conduct the group pursuit. That means we do not know the group structure, but we use the pairwise network structure to see that if we can capture this group structure. So here, the V has an even more clear group structure, so it contains 4 groups. If you apply the L1 sparse CCA, you won't capture the first group. But if you apply the network structure's sparse CCA, so we successfully capture all the 4 groups here. So now we apply the structure sparse CCA to real eQTL mapping data. So basically, it's the EAST data. So there are about 124 samples, about 1,000 genes and 1,000 SNPs. The SNPs are from the chromosomes 1 to 16. So we extracted 92 pathways from the KEG dataset. So we construct, like you can view that, we construct 92 groups of these genes. And the largest group contains about 475 genes. So the average group size is about 25. And the groups are highly overlapped together. I highlight again. So each gene on average appear in about 2 groups. So we conducted the L1 sparse CCA and the group structure sparse CCA. So the group is extracted from the KEG database. So for the L1 sparse CCA, we select about 71 genes and about 50 pathways. For this one, we select much less genes, about 47 genes with the 32 pathways. And we conduct a functional enrichment on this for both L1 sparse CCA and the group sparse CCA. So I cannot put the full list of the functional enrichment here, but basically the group sparse CCA leads to much smaller p value, and it's characterized a few important functions of the yeast, like the tyranoid backbone biosynthesis. And also another interesting observation is that we put the structure penalty on the V side, but it can also affect the variable selection on the U side. That means if we do the group structure on the genes, but it's also affect the variable selection on the SNP. So here is some interesting observation. If you use the L1 sparse CCA, you get this 173 SNPs, but across almost every chromosome. But use the group sparse CCA, you get 121 SNPs, but they're concentrated on the 12th and 13th chromosome. And now we are collaborating with some biologists to further verify this, carry out some more meaningful biological observation. This is all the SNPs here are concentrated on the chromosomes 12 and 13. And also finally we apply this network structure sparse CCA, and we find that this is the selected genes, and we can easily identify these 2 groups, and we can also do some enrichment, the functional enrichment on these 2 groups, and leading to some meaningful biological observation. So the code is available on my website, so for those who are interested, you can directly download the code. And also for any feedback, please send to my email. So thank you very much.
Time for questions. Well, ask,
sure.
At the beginning, you justified the approach, saying there's a symmetry between the X's and the Y's.
Yeah.
The X's representing the genome, the SNPs, and Y's representing the expression values. But from the biology, I mean, there's a directional flow of sort of causality, so that the X's, the variation at the genome level, cause changes at the transcription level. So, I mean, sort of SNPs might be indicative of something that happened at the genome level that actually causes a change of the Y's. So for that reason, I think most of the approaches have used regression so far. So how can you just defend again why you are so critical of the regression approach and think that it should be treated as a symmetric.
Oh, I see. It's a very good question. So I know that recently Peter Buhlmann has done some experiment, and he has a very nice paper to say that if you want to find the causality relationship, but if you do the simple regression, you cannot easily capture this causality. So that means causality cannot be easily captured. That, for example, you just use ASO procedure, the improvement is very limited. So that if you want to really capture the causality, it's not mean that you can simply do the procedure by ASO and select those variables. It's not the right way to do the causality. So that means we think that we are more interested in finding the association between the SNP and the gene expression data, so that we resort to the canonical correlation analysis approach.
Empirical comparisons with sparse regression?
Yeah, but here since we select both the SNP and the genes, but for the sparse regression, it only select the SNPs. So it can be done with you select a few SNPs, but trying to explain all the gene expression data. So basically, I think they are kind of still different. So I haven't conducted a real experiment to compare that.
Any more questions? We'll have another question. I mean, you sort of my understanding was you have two different methods, the group sparsity and the group pursuit method.
Yeah.
So what is the difference? In one case, you've got a hardwired network. In the other case, you bring in these prior knowledge variables W. So how slack are they?
So basically, they are for two different purposes. In some cases, if you know your group structure, for example, you extract the group structure from your pathway, you can incorporate this group structure information. So it's natural to use, and you do the group-wide selection. So you use this one. But in some other cases, you do not know your group structure, but you certainly do not want to study like the functional enrichment of your whole selected genes, which could lead to a very messy results. You want to separate them into small different groups. So that's why I think you can first apply the group pursuit. You can classify them into different groups, and then maybe you can select your meaningful groups. So the second way is that in the case that you do not have a prior knowledge of the group structure, and you use this kind of to learn the group structure.
So if there are no more questions, let's thank the speaker again.